Welcome back, everyone, to the deep dive. Really excited about today's exploration. We're diving into, well, a fascinating, maybe even revolutionary idea about aging itself. Forget everything you thought you knew, maybe, about a single aging gene or just wear and tear. What if it's more like a demented pianist messing up the beautiful music in our cells? That's the mystery we're unpacking.
Exactly. And to really get our heads around this, we're going to trace the story. We'll start with the Human Genome Project, the initial map and this whole idea of junk DNA. Then we'll detour into the world of, believe it or not, yeast research, a tiny organism with big answers. And finally, we'll land on this big idea, the information theory of aging. And yeah, we'll even touch on whether we can actually reverse aging. It's quite a journey.
Okay, let's rewind then. April 15, 2003. Big news. The human genome is complete. But that wasn't quite the whole picture, was it? There were still gaps.
Yeah.
Pesky gaps.
Right. And it wasn't scientists like overpromising. It was really about the technology we had back then. They felt they'd gotten as close as they possibly could to nailing down all 3 billion base pairs. That was the limit.
And these missing bits, they were often these repetitive chunks of DNA. Dismissed as junk, right? Like old viral leftovers or something. People thought, okay, we got the important stuff, the genes that make us us.
Yeah, that was the thinking. But this so-called genetic dark matter, this junk, turns out it's up to 69% of our genome. Huge. And even in the parts they thought they had decoded, maybe 10% was still missing, including areas we now know are really important for aging. Plus the early computer programs, the algorithms, they were set up to ignore small genes, anything under 300 base pairs. But genes can be tiny, like 21 base pairs long. So thousands of really essential bits were just missed.
Wow. Okay, so if we miss so much, what does that mean for finding, like, the aging gene?
Well, here's the twist. It actually suggests we won't find a single aging gene. Think about it. Evolution wouldn't select for a gene whose sole purpose is to make us deteriorate. It doesn't make sense. We definitely have genes that influence how long we live, longevity, genes that control our defenses, and genes that affect the symptoms of aging. But one specific gene designed to cause aging, like, say, an oncogene causes cancer, probably not. Aging seems to be something else.
Okay, so not a specific gene, but maybe a process. And understanding that process, surprisingly, involved yeast. Tell us about Robert Mortimer.
Ah, Mortimer, yes. A key figure, maybe a bit unsung. He was visionary, kind. His big contribution was championing Saccharomyces cerevisiae, that's baker's yeast, the sugar-loving kind, as a serious research tool. He gathered thousands of different mutant strains, and crucially, he made them available to anyone, anywhere, through the Yeast Genetic Stock Center. You just paid postage.
Just postage. That's amazing. So it made yeast research accessible. Because I imagine people were skeptical at first, right? Using yeast to study human aging.
Oh, definitely skeptical. But yeast cells, despite being separated from us by like a billion years of evolution, are surprisingly complex. Genetically, biochemically. They share about 70% of our genes. Their basic life processes, eating, reproducing, responding to stress, are pretty similar. And the key thing for aging, they age incredibly fast. A whole lifespan in a week.
A week. Okay, I see why that's useful. Fast turnaround.
Exactly. Ideal for studying aging. It's why yeast genetics has led to, I think, 5 Nobel Prizes, including the one in 2009 for figuring out telomere shortening.
And the author of the book we're drawing from, he had a personal connection here, met Mortimer.
He did.
Yep.
Met him in Vienna in '92. That meeting and conversations with others like Leonard Guarente at MIT really fired him up about studying aging in yeast, Guarente especially. They kind of made a pact. If we can't figure out aging in this simple yeast cell, what chance do we have in humans? It became his mission.
And there's a story about him getting into Guarente's lab, something about a bottle of wine.
Yeah. He was persistent, a foreigner trying to get a top U.S. fellowship. He apparently gave Douglas Melton, who was interviewing him, a bottle of Aussie wine. He worried later it looked like a bribe. But he got the fellowship, the Helen Hay Whitney. A life-changing moment, really. Set him on this path.
So, fellowship secured, lab position ready. The initial plan was to study Werner syndrome, that rare disease causing really rapid aging.
That was the plan. People with Werner's, they start showing signs of old age in their 30s or 40s, only live to about 46. A tragic model of accelerated aging. But here's the irony. Literally within 2 weeks of the author starting, a team at the University of Washington scooped them. They found the WRN gene that causes Werner syndrome.
Oh, wow. That must have been deflating.
Totally deflating initially. But it actually turned out to be the key that unlocked the information theory of aging. Because the next obvious step was, does the yeast version of this gene, SGS1, do the same thing.
So they decided to test it. Give the yeast Werner syndrome, basically.
Pretty much. They engineered yeast cells, swapping out the normal functional SGS1 gene for a broken mutant version.
And the result?
Dramatic. First, their lifespan was cut in half. But it wasn't just that they died sooner. They aged faster. Their cell cycle slowed right down. They got physically larger. And weirdly, they became sterile.
Sterile? Why sterile?
Because both their mating type genes, think male and female identities, got switched on at the same time. They lost their specific cellular identity. It looked exactly like a yeast version of Werner's. And visually, using special stains, they could see it. The nucleolus, this dense spot in the nucleus where ribosomal DNA is kept, it looked like it had exploded in the aged mutant cells, scattered into little islands. The author described the image, which ended up in Science, as tragic and beautiful.
Tragic and beautiful. So what was happening? What caused this explosion and loss of identity?
The key was a protein called SIR2, the very first sirtuin discovered. It's what we call an epigenetic factor. Normally, it hangs out near the mating genes, keeping them quiet. But when the nucleolus got damaged in these mutant yeasts, SIR2 moved. It left the mating genes and went into the damaged nucleolus, presumably to help with repairs.
So it abandoned its post.
Essentially, yes. SIR2 works by bundling up DNA, removing little chemical tags called acetals from proteins called histones, which keeps genes silent. When it left the mating genes, they weren't silenced anymore, and the cells lost their sexual identity.
Okay, so broken DNA in one place causes SIR2 to move, which messes up gene silencing somewhere else, leading to aging symptoms.
Exactly. The author wrestled with this for weeks. Then, apparently, in the wee hours of October 28, 1996, it clicked. The core idea. He called it wisps of a concept at first. Broken DNA causes instability in the genome. This instability distracts proteins like SIR2. SIR2 moves, changing the epigenome, how genes are controlled, cells lose their identity, become sterile, malfunction, all while the cell is trying to fix the initial DNA damage. That was it. Epigenetic changes cause aging. It wasn't just mutations, it was this information loss.
Wow. And what did his mentor, Guarantee, say?
Classic understated scientist response. 6 words. I like it. Go prove it.
Go prove it. Okay, challenge accepted. So let's dig into this epigenome a bit more. It's not the DNA sequence itself, but how it's managed.
Precisely. Think of your DNA as the hardware, the raw code. It's wrapped around these protein schools called histones, then bundled up further into chromatin and finally chromosomes. Sirtuins, like SIR2, are part of the software. They tell the histones how tightly to wrap the DNA. Tightly wrapped means genes are off. Silence, that's called heterochromatin. Looser means genes are accessible, ready to be read euchromatin. Sirtuins do this by removing those acetyl tags, basically telling transcription factors, nope, you can't read this part.
So every cell, skin, nerve, liver has the same basic DNA blueprint. But the epigenome acts like a director, telling each cell which parts of the blueprint to read and which to ignore. That's what makes them different.
You got it. The epigenome controls cellular identity and function. It dictates huge parts of our lives, far more than we used to think. The analogy the author uses is great. Think of the genome as a grand piano. All the keys are there, fixed, stable. That's your DNA sequence, the epigenome. That's the pianist. A virtuoso, playing the keys softly, loudly, holding notes, playing quickly, creating the unique music of each cell type.
I like that. Piano and pianist. So DNA isn't destiny then, that old idea.
Not nearly as much as we thought. Look at identical twins.
Thank you.
Same DNA, right. But if one smokes and the other doesn't, they age very differently. Their epigenomes diverge. Studies suggest our genes only account for maybe 10 to 25% of our lifespan variation. The rest, environment, lifestyle, and how they impact the epigenome.
Okay, so the pianist is key, but what happens when things go wrong? This demented pianist idea.
Right, so imagine that virtuoso pianist starts slipping, missing keys, hitting wrong notes, playing sections that should be silent. The piano itself, the DNA, is still perfectly fine. All the keys are there. But the pianist is becoming, well, unhinged. The music turns into noise.
And that noise is aging. The gray hair, the wrinkles, the aching joints. It's the epigenome messing up the performance.
That's the core of the information theory of aging. This epigenetic noise, largely triggered by the cell constantly dealing with disruptive insults like DNA breaks, just like in those yeast cells, is the fundamental reason we age. It's not just one thing. This noise drives all the classic hallmarks of aging we talk about. Stem cells getting exhausted, cells becoming senescent, mitochondria malfunctioning, telomeres shortening. They're all downstream consequences of this epigenetic chaos. It's a unifying theory.
And they found more proof back in the yeast, beyond just seeing SIR2 move.
Yes. Using a technique called a southern blot, they looked closely at the ribosomal DNA that's stuffed in the nucleolus that exploded. In aging yeast, both of Werner mutants and normally aging yeast, this rDNA was madly unpacking. It was forming these little rogue circles of DNA outside the main chromosomes. They called them extrachromosomal ribosomal DNA circles, or ERCs. And these ERCs piled up as the yeast got older.
Okay, so these ERCs show up during aging, but are they just a symptom or are they causing it?
That was the million-dollar question. So they did a clever genetic trick. They basically forced these ERCs into young, healthy yeast cells. And boom, premature aging, sterility, early death.
Wow. So the ERCs themselves caused aging.
Exactly. That was the smoking gun. Published in Cell in late 97. And then another researcher, Matt Kaberlein, did the flip side. He gave yeast an extra copy of the SIR2 gene. Those yeast made fewer ERCs and lived 30% longer.
Incredible. So it all fits together. Genome instability like DNA breaks distracts SIR2, leading to epigenetic changes like ERCs, which causes aging.
Right. The sequence looks like this. Youth. Inevitable DNA breaks happen. Leads to genome instability, which disrupts DNA packaging and gene regulation. That's the epigenome going haywire. Cells lose their identity. They become senescent or malfunction. Disease risk goes up. And eventually, death.
That's a remarkably clear picture.
And the implication is huge. If you can intervene at any point in that cascade, maybe you can slow aging. What if you could intervene at multiple points? Could you actually stop it? Or even reverse it?
Which brings us to this survival circuit. You mentioned our DNA is under constant attack. Trillions of breaks a day.
Yeah, it's mind-boggling. Every time a cell divides, chromosomes can break, plus damage from UV light, toxins, just metabolic byproducts. It's constant. So over billions of years, life evolved a fundamental response. Sense the damage, pause growth and reproduction, and divert all available energy to fixing the breaks. It's a deeply ingrained survival program.
And we see this in mammals, too, not just yeast, with the mammalian versions of SIR2.
Absolutely. We have SIR1 and SIR6, direct descendants of yeast SIR2. They are critical epigenetic controllers, and they're also recruited to DNA repair sites. If you engineer mice to lack SIR1 or SIRT6, their DNA repair is faulty and they age much faster.
But the flip side also holds true. Giving them extra sirtuins helps.
It does. Just like adding extra SIR2 boosted yeast lifespan, giving mice extra copies of SIRT1 or SIRT6 makes them healthier and extends their lives.
So the analogy you used earlier, the disaster response corps, sirtuins rushing off to fix DNA breaks.
Exactly. Think of sirtuins like the utility workers or emergency crews in a city. They have their regular jobs keeping the power grid stable, the water flowing. That's gene regulation, maintaining cell identity. But when there's an emergency, a water main break, a downed power line, a DNA break, they have to rush off to fix it.
Okay, but here's the crucial part you mentioned. If the emergencies are constant, the crews are always busy elsewhere, and they don't always go back to their original posts afterwards.
That's the problem. They get distracted. They might fix the DNA break, but then they don't always find their way back to the exact gene they were silencing before. Or maybe they're slow eating back. And the result? Genes that should be off might flicker on. Genes that should be on might get silenced. The cell's carefully orchestrated gene expression pattern gets scrambled. Cells lose their identity, start malfunctioning. It's chaos. That's epigenetic noise, pure and simple. Aging.
And the reason this happens is that cells just don't have enough sirtuins to do both jobs, routine maintenance and constant emergency repair simultaneously.
Precisely. They exist in limiting amounts. There aren't enough SIRT1 molecules, for instance, to sit permanently on all the genes they need to silence and be instantly available to rush off to every single DNA break, so they shuttle back and forth.
Which raises a question. If having more sirtuins is better for longevity, why didn't evolution just give us more?
Ah, the evolutionary paradox.
Yeah.
Because making extra proteins costs energy. In the wild, resources are scarce. A slightly longer lifespan might not be worth the extra energy cost if it means you reproduce less or can't survive a famine. Evolution optimizes for reproductive success in a harsh environment, not maximum lifespan in a cushy lab with unlimited food, but in the lab. Give them extra SIR2 or SIRT1, remove resource limits, and boom, longer life.
Okay, so the theory is solid. DNA breaks distract epigenetic regulators, like sirtuins, causing epigenetic noise, which is aging, but proving this definitively in a mammal. How do they do that? They needed to cause just the DNA breaks without mutations, right?
Exactly. They needed a really precise tool, and they found one, amazingly, in a slime mold called Physarum polycephalum. It makes an enzyme, IPPTI. This enzyme is incredible. It cuts DNA only at a very specific long sequence that barely exists in the mouse genome. And it makes clean cuts, no mutations. The cell's repair machinery just neatly pastes the ends back together. A perfect tool for causing temporary breaks. A nature's gift to science, as the author calls it.
So they use this enzyme to create special mice, ICEs.
Right. ICE stands for inducible changes to the epigenome. They put the gene for this IPPI enzyme into mouse embryonic stem cells, then made mice from them. These mice were born completely normal, and this is the inducible part, they engineered it so the IPPI gene would only turn on when the mice were given a low dose of tamoxifen, the drug used in breast cancer treatment.
Okay, so tamoxifen flips the switch, the enzyme starts making clean DNA breaks, sirtuins get distracted, and then if you stop the tamoxifen, the cutting stops.
Exactly. Controlled epigenetic disruption. The hypothesis was giving these mice tamoxifen for a few weeks would accelerate the aging process just by causing this epigenetic noise without causing mutations or other damage.
Like a Harry Potter aging potion. Did it work?
It worked like wizardry, apparently.
Huh?
For months after the tamoxifen treatment stopped, the mice looked fine. Then one day, a postdoc emails the author, worried about a mouse that looks really sick, wants to euthanize it. The author sees the photo and says he just laughed. He told the postdoc, "That's not a sick mouse. That's an old mouse."
No way. What did it look like?
At just 16 months old, which is like midlife for a mouse, maybe 65 in human years, this ICE mouse that got the tamoxifen pulse looked ancient. Thinning, graying fur, hunched spine, ears like paper, cloudy eyes, classic signs of old age. Meanwhile, its siblings, same age, same genetics, but no tamoxifen, perfectly healthy and youthful.
That is incredible. And crucially, they hadn't messed with the actual DNA sequence. No mutations. Didn't target telomeres or mitochondria directly?
Nope. They only induced temporary DNA breaks, mostly in the non-coding junk regions. But that was enough. Enough to trigger the sirtuin response, shift their locations, scramble the epigenome, alter gene expression patterns across the board, and induce all the symptoms of aging. They even measured their biological age using the Horvath clock, an epigenetic clock. The ICE mice were biologically about 50% older than their untreated siblings. They'd found, as the author put it, life's master clock winder.
It's like scratching a DVD. The digital information, the DNA, is still perfect. But the analog player, the epigenome reading it, gets damaged and can't access the information properly.
Perfect analogy. And the conclusion was inescapable. Aging, at its core, seems to be caused by this loss of epigenetic information triggered by the cell's response to DNA damage.
Yeah.
They hadn't given the mice wrinkles and gray hair, then frail bones individually. They'd given them aging.
And the implication, if you can give aging,
Then maybe, just maybe, you can take it away.
wow. Okay, that is profound. It makes you think about organisms that naturally seem to defy aging, like those ancient bristlecone pine trees,
Yeah.
thousands of years old.
Right. They show what scientists call negligible senescence. Basically, no signs of aging. Their cells don't seem to decline or accumulate mutations over millennia. And there are others. The little freshwater polyp, Hydra vulgaris. They've kept them in labs for over 40 years with no signs of aging.
And the immortal jellyfish.
Yeah, Turritopsis dohrnii. It can revert back to a polyp stage when stressed, basically resetting its aging clock. And the Greenland shark, living over 500 years. The bowhead whale, one found that was over 200 years old.
Incredible lifespans. But genetically, they're still related to us, right? They aren't complete aliens.
Not at all. We share 70% of our genes with yeast, remember? And we share almost 13,000 genes with the bowhead whale. There's a lot of common ground. One key shared pathway involves a family of genes called FOXO or OF16 in worms, MSN2 in yeast. These are master regulators involved in stress resistance. They turn on defense programs, repair damage, and they're intimately linked with sirtuins and NAD, the fuel sirtuins use.
And these FOXO genes are relevant in humans, too.
Very much so. Certain variants of the FOXO3 gene are found much more frequently in people who live exceptionally long lives, like centenarians. It seems these variants keep our cellular defense and repair systems more active throughout life, protecting against age-related diseases. You can even check your own genome for these variants nowadays.
Interesting. So we all have this basic survival circuit, this machinery involving sirtuins and FOXO, and we're all subject to DNA damage and epigenetic noise, yet a yeast lives a week, a human maybe 80 years, a whale 200, a jellyfish potentially forever. How do they manage the noise so differently?
That's the big question. How does a whale keep that epigenetic symphony playing clearly for centuries? How does a jellyfish hit the reset button? This leads back to the idea of the epigenetic landscape.
Right, Conrad Waddington's idea from the 50s, the marble on the mountaintop.
Exactly. He visualized a developing embryonic stem cell as a marble perched at a peak. As it differentiates, it rolls down into a specific valley, becoming a nerve cell, or a skin cell, or a liver cell. The landscape, shaped by the epigenome, guides the marble and keeps it in its valley, maintaining its fate.
But you said earlier there's no fixed fate in biology.
Right. Waddington's original model implied the valleys were deep and the marble stayed put. But the reality, especially with aging, is different. Think of those DNA breaks as little earthquakes. They shake the landscape. Over time, with enough shaking, the valley walls erode, the marble starts to wobble. The cell X-differentiates, it loses its precise identity. A skin cell might become, say, 90% skin cell, but 10% acting like other cell types. It becomes confused, inept. That's the cell succumbing to epigenetic noise.
Because the epigenome is like analog information, prone to getting degraded over time.
Exactly. Unlike digital DNA code, which is pretty robust, the epigenetic layer, the histone markings, the DNA methylation patterns, is more like an analog recording. It can get scratchy, lose fidelity. Evolution built us to reproduce, not necessarily to live forever, so the valley walls aren't infinitely high, the gravity holding the marble isn't infinitely strong. And the primary cause of this erosion, according to the theory, is that constant shuttling of epigenetic factors like sirtuins. Away from genes to fix DNA, then back again. But maybe not perfectly back, leading to the wrong genes turning on or off at the wrong times. The demented pianist strikes again.
Just like inducing those DNA breaks in the ICE mice caused the landscape to erode faster, making their cells confused and leading to aging.
Precisely. Their bodies became, as the author puts it, chimeras of misguided, malfunctioning cells. They got aging because their epigenetic information was degrading.
So, if information loss is the core problem, can we restore the information? Can we make those valley walls higher, strengthen the gravity, push the marbles back where they belong?
That is the billion-dollar question, and the answer seems to be yes. The author says, I can say with confidence that there is.
Okay, that's a bold claim. Reversing aging, what's the evidence?
Well, remember those ICE mice? They did the reverse experiment too. But even before that, there was this anecdote from the lab. They had some old mice, 20 months old, so like 65 in human years. And the postdoc running the experiments had a problem.
A problem? What kind of problem?
The mice won't stop running. That was the problem.
They won't stop running. Old mice.
Yeah, they had apparently broken the software on the mouse treadmill because it wasn't programmed for mice running that far over 3 kilometers. These elderly mice were running ultramarathons.
Okay, what was going on with these super mice?
They had been treated with an NAD-boosting molecule. NAD is the fuel sirtuins need to work. So boosting NAD effectively activated their SIRT1 enzyme. And what happened? The activated sirtuins prompted the cells lining their blood vessels to grow new capillaries into their muscles. More blood flow, more oxygen, better waste removal. It reversed age-related frailty. It acted like an exercise mimetic.
So boosting sirtuin activity made old mice physically younger, more energetic.
It certainly seemed to reverse key aspects of aging. In terms of the landscape analogy, activating sirtuins was making their epigenomes more stable. The valley walls were getting higher, the gravity stronger. Waddington's marbles were being nudged back towards their youthful state.
That's incredible. A real sign that age reversal, at least some aspects of it, is possible.
Definitely. There are still questions, of course, which molecules, what doses, long-term effects. But the fundamental principle that epigenetic information loss drives aging and restoring it might reverse aging seems to hold.
And the exciting part is we don't necessarily need to wait for miracle drugs. Things we can do now, diet, exercise, managing stress likely work. In part, by stabilizing our own epigenomes, we can already take steps.
Absolutely. There's a lot we can potentially influence already. But before we dive into all the potential interventions, the life extension strategies, the treatments on the horizon, there's that really big question we need to face.
Right.
Should we?
Wow, what a deep dive today. We've covered so much ground. From realizing the human genome map wasn't quite complete and the importance of junk DNA to the crucial role of simple yeast in figuring out complex biology and landing on this powerful information theory of aging. Aging as epigenetic noise triggered by DNA damage response, and then the ICE mice actually inducing aging just by scrambling that information.
It's a compelling story. Aging is information loss.
Yeah.
But the flip side is the hope. If information can be lost, maybe it can be restored. If we can cause aging, maybe we can reverse it.
Which brings us right back to that final provocative question you posed. Should we? If we gain the power to dramatically extend lifespan or even reverse aging, Should we use it? What are the consequences? It opens up a huge discussion, doesn't it? Ethics, society, what it means to be human. Well, thank you for joining us on this fascinating journey today. We hope it's given you a lot to think about. Join us next time as we start to unpack the answer to that very question, should we, and explore what science is already revealing about how we might slow, stop, and yes, maybe even reverse the aging process.